I want to thank the webinar hosts for inviting me to speak here today and also to express my deep appreciation to my two good friends, Dr. Werner Seeger and Sir Magdi Yacoub, for agreeing to join me during this session. I have been working and always full of admiration with Werner and Sir Magdi since the 1990s. Werner has been my guide through therapeutics, and Sir Magdi has been my guide through organ manufacturing. I feel extraordinarily lucky to have had continuing opportunities up to the current year and beyond to learn from these two magnificent scientists and physicians. The title of my talk is A Personal Journey Through PAH and Its Treatment Options. It is a personal journey because I am the parent of a child with PAH. I have been trained as a medical ethicist, not a pharmaceutical scientist or a medical doctor. I am also a technologist and one who is technologically agnostic, meaning I don't really care if a technology is invented by me or someone else. As long as it works best, that is the technology I want to use. I like the Chinese proverb that it doesn't matter if a cat is black or white, so long as it catches mice. T.S. Eliot explains well how I feel about PAH. I have been leading teams exploring it and its treatment options for over a quarter of a century, and while we are actually far from the end of our explorations, at this point we have kind of arrived back where we started, with the parenteral prostacyclin mostly known as Remodulin. However, while I first thought of Remodulin as a long-term destination, I now know it as but a short-term bridge. The misunderstanding of it as a long-term destination came from extrapolating from the results of Floland's mortality improvement outcome. The new theory of it as a short-term bridge comes from recent retrospective data analyses showing that if dosed aggressively to bring a patient to mean pulmonary artery pressure under 40 milligrams of mercury, the patient could then be transferred to an easier oral therapy. But knowing and proving something is not the same thing. This is what my personal journey through PAH and its treatment options has taught me. My mission now is to prove it, which we intend to do in our new Artisan clinical trial. So that I don't get too far ahead of myself, let me back up and explain how my explorations wandered over the years, what wonderful new therapies resulted, and why I think that I now know this place called PAH, or at least Remodulin, for the first time. When I started my journey, the average survival was only 3 years. Today, it is more than twice as good, but clearly we still have a long way to go. My journey has been inspired by the story of these two remarkable people, Sir Magdi Yacoub and Dame Julia Pollock. Dame Julia was a histopathologist working with Sir Magdi, one of the greatest transplant surgeons, when she developed PAH. Medicines couldn't help her much, but a heart-lung transplant from Sir Magdi gave her 15 years. That all taught me that I must fight a two-front war, better medicines on the one hand and enough transplants for all on the other hand. I started to increase the supply of transplants by restoring donated but unusable lungs to transplantability. Previously, 80% of donated lungs were discarded and unusable. Now, we are able to restore more than half of those to transplantability. The following video will show you how we do it. This technology has already made a big increase in the availability of lung transplants.
Through our lung service model, United Therapeutics currently receives lungs from transplant centers east of the Mississippi River. Our LB1 facility in Silver Spring, Maryland is the first full-service ex vivo lung perfusion center in the U.S., housing 6 EVLP procedure rooms. Once the lungs arrive at our facility, our CMX system automatically sends a notification to the participating centers, including the OPO, the transplant center, the LB1 clinical staff, and the medical advisory team. The UNOS number and blood type are verified in CMX, and a back table evaluation commences noting organ condition. Perfusion takes place over a 3 to 6 hour period, during which the donated lungs are placed into a sterile plastic dome and attached to a ventilator, pump, and filters. The lungs are maintained at normal body temperature and treated with a bloodless solution that contains nutrients, proteins, and oxygen. Throughout this process, ongoing CMX documentation and assessment steps take place. These include bronchoscopy, X-rays, and blood gas analysis. During this period, our IT specialists facilitate visual and data communication with the transplant centers. Through our EVLP process, we are working to reduce the devastating effects of end-stage lung disease.
This picture shows me in a white suit with Heather Leverington, the recipient of 2 of our restored lungs. Heather was a college sports coach and then got an aggressive case of PAH. No medicines could save her. That is why we are working hard to create better medicines. But our restored lungs worked so well for her that she won the gold medal in shot put at the U.S. Transplant Games. This shows why we must fight a 2-front battle: better medicines and an unlimited supply of tolerable transplantable organs. When my daughter Jenesis was diagnosed with PAH, then called primary pulmonary hypertension, or PPH, in the early 1990s, there were no approved treatments. As a satellite communications technologist, I didn't know what to do. It was 1992, and I had just seen a movie called Lorenzo's Oil, starring Nick Nolte, about a father who tried to save his young son from a fatal illness with a concoction disguised as salad dressing oil. So I began an intensive personal research effort to produce a vasodilating nutraceutical, what Forbes magazine called Jenny's oil. It didn't work, perhaps because its arginine-loaded contents tasted horrible. Patient compliance is often our worst enemy. She got rapidly worse, with syncopic episodes at home and at school, dyspnea all the time, cyanotic lips, and chest pain near her ever-expanding right heart. We took her to Children's National Medical Center in Washington, D.C., the top source of relevant expertise in the mid-Atlantic where we lived. She was given a grim prognosis after cardiac catheterization with virtually nothing to hope for. Fortunately, Jenesis was a calcium channel blocker responder, but the head of pediatric cardiology said that it would not last, and she probably had just a few years to live and then would need a lung transplant. At this point, it was literally do or die. The company of my dreams that I had created, Sirius Satellite Radio, had just gone public. So, my partner Bina and I decided to throw all that money and all of our time into finding a cure for Jenesis before she ran out of time. All we could think of doing was starting a nonprofit foundation and giving grants to companies and doctors working on a cure for PAH, while self-teaching ourselves everything there was to learn about pulmonary artery physiology and biochemistry. I got a list from the Pulmonary Hypertension Patient Support Group of all the doctors working on the disease in the United States. There were about 30. The only company working on it was Burroughs Wellcome. We sent each of them a request for proposals for funding of a treatment that would be available for Jenesis in under 3 years. We received responses from Dr. Rubin, Dr. Barst, Dr. Vocal, and a couple of others. While we funded them, only 2 good things came out of the effort: knowledge that no patient who received a lung transplant ever redeveloped PAH, and an introduction to Dr. Robin Barst. Meanwhile, as Jenesis spent ever more time as an inpatient, hooked up to Holter monitors at Children's, I spent ever more time in their basement medical library. In that library's print journals, I learned of a compound that had been tested for congestive heart failure and failed, but that showed strong pulmonary selective vasodilation. By then, I knew that was exactly what I needed. I was seeing a glimmer of hope on 2 counts. I started working on 2 things: medicines to help Jenesis live longer, and a way for her to have a lung transplant without rejection. I also moved Jenesis to Dr. Robin Barst, even though she was far away in New York City, because she was the only respondent to my grant proposals that claimed expertise in pediatric pulmonary hypertension. The first thing Dr. Barst did was to increase tenfold my daughter's dosage of nifedipine. The dose was so high that it was always a great challenge to persuade the pharmacies that the dosage was not an error. Yet, I believe there was a peek into the future in her practice. The aggressive dosing of nifedipine drove way down Jenesis's pulmonary artery pressures, and she began to do much better. Dr. Barst also introduced me to Dr. James Crow, who in turn introduced me to Sir John Vane when I told her I needed help getting this new treprostinil molecule that I had found in publications at Children's Hospital developed for PAH. She was also aware of the molecule and was annoyed it had been discontinued by Glaxo after it purchased Burroughs Wellcome. She said my getting that molecule developed was the very best thing I could do to help my daughter. She also said Doctors Crow and Vane could help me get it out of Glaxo because Dr. Crow and Sir John used to work at Burroughs Wellcome. Additionally, Sir John was a Nobel laureate in the field. When I met Sir John, he agreed to help me get the molecule out of Glaxo, and he also agreed to help me pursue my plan for a supply of tolerable xeno lungs by recommending me for a PhD in xenotransplantation studies at the St. Bart's and Royal College of Medicine and Dentistry at the University of London. Most people don't like to look medicalized, and this is probably more so for young teenagers. Noncompliance is a big problem with immunosuppressants, and high doses of nifedipine cause gingival hyperplasia. We felt we had to find a better solution, especially as our daughter's mean pulmonary artery pressure was creeping back up. The solution that I thought we had, subcutaneously delivered treprostinil, now appeared chimerical. The rights were purchased from Glaxo, but they had no useful drug to give us. Nobody in the U.S. knew how to make it, and nobody knew how to deliver it continuously. I felt that we had gone through an enormous amount of effort with nothing to show for it. But finally, after a year of relentless door-knocking and cajoling, we were able to get the head of chemistry at University of Illinois, Dr. Robert Moriarty, to try and synthesize it, and we were able to get the head of an insulin delivery pump company, Dr. Al Mann, to try and modify his device to work with treprostinil. Meanwhile, so that all of our eggs weren't in the treprostinil basket, Sir John introduced me to Torii and Asahi in Japan, from whom I licensed 2 other prostacyclin analogs, Bariprost and AFP007, while Dr. Seeger introduced me to Schering AG, from who I valiantly tried but failed to license Cicaprost. But a 4th analog, Elaprost, was making good progress in Dr. Seeger's hands. By the turn of the century, there was a light at the end of the tunnel, but it was a problematic light in many ways. While parenteral therapies like Flolan and Remodulin could reduce mean pulmonary artery pressures if dosed aggressively enough, they required wearing a pump for the rest of one's life and exposed a patient to sepsis and rebound hypertension risks if taken intravenously, and to monthly infusion site pain if taken subcutaneously. Oral drugs such as bosentan and sildenafil were on the horizon. The Kaplan-Meier curves showed a steady rate of death from patients on those therapies. It is very hard for a parent to tell their child that they can't go to the beach or swim, as is the case with Flolan or intravenous Remodulin, or that they have to have a catheter coming out of their skin. Of course, these are heavy burdens for any patient, whatever their age. It is true that patients can adapt to all kinds of difficult therapies, but that doesn't make it pleasant. Humans can adapt to all kinds of hardships, but they are counting on us to help relieve those hardships as soon as possible. How nice would it be to say to a patient, "You may only have to put up with this parenteral treatment until you are considered hemodynamically stable, and then you can transition to a pill." I had no idea such a protocol was possible in the early 2000s. If we did, I do not believe we would lose to death approximately 40% of PAH patients who succumbed to the disease without ever trying parenteral prostacyclins due to the fear that the treatment was worse than the disease. Humans can adapt if they see hope instead of an endless tunnel. In our case, my daughter pleaded with me to create an inhaled treatment for her PAH so that she would be no different than her friends with asthma inhalers. Although many insisted that treprostinil, the drug that we finally made work subcutaneously and intravenously and branded as Remodulin, would never work as an inhalation, Dr. Seeger saw things differently and proved to me scientifically that it would work. At that point, I directed my efforts to creating an inhaled version of Remodulin that would ultimately be called Tyvaso. To solve the problems of the indwelling catheter and sub-Q site pain, we next developed Tyvaso, an inhaled form of treprostinil. This solved the problems of parenteral delivery. I also think it is a bit larger than my daughter's friends' asthma inhalers. Nevertheless, this Tyvaso inhaled product allows patients to unplug from the pump and has the potential to give them a lot of their personal freedom back. I'm sure just the availability of Tyvaso has persuaded many patients to go onto the parenteral pump for a while with the hope that they can be transitioned to a less invasive, longer-term maintenance therapy. But we are relentless in making life ever better for our patients. So, we are now in the process of replacing this device with a much smaller treprostinil inhalation device. In fact, we filed for FDA approval of it a few weeks ago. I think we finally have a chance to get my daughter her asthma inhaler. When this device is approved, hopefully in December of this year, it should be able to be used not only for WHO Group 1 PAH, but also for the WHO Group 3 PAH segment, PAH-ILD, largely patients with pulmonary hypertension associated with interstitial lung disease, in addition to pulmonary hypertension. We received FDA approval for the WHO Group 3 patients just last month. This is an example of if you do the right thing with the right intentions, you can reap unexpected rewards. As noted earlier, the goal of us developing Tyvaso was to be able to provide benefits of prostacyclin therapy in a more convenient formulation. However, another benefit was that we were able to demonstrate improvement in clinical outcomes without inducing ventilation perfusion mismatch of VQ mismatch. This means that we had a unique ability to help patients with WHO Group 3 PAH. It is sometimes said that the road to hell is paved with good intentions, but I believe it is just as true, and in fact more so, that the road to heaven is also paved with good intentions. A further benefit of addressing WHO Group 3 patients is that our secondary endpoint suggested that, when inhaled, treprostinil also may have antifibrotic properties because there was marked improvement in FVC in the treated compared to the control group. We are now conducting a study specifically in IPF patients without PH called the Teton study. And while we are conducting the study with the larger Tyvaso nebulizer, I believe we will be able to offer those patients, if and when the shown Dreamboat device is approved for Tyvaso, and if we are successful in our Teton study of Tyvaso in IPF, this finger-sized device to help treat their IPF. Furthermore, I believe our entry into PH Group 3 pulmonary hypertension may help these additional patients better manage their disease. As you can see from these Kaplan-Meier plots, the survival of ILD patients with pulmonary hypertension is much worse for those patients without PH. Between the new approval of Tyvaso for ILD patients with pulmonary hypertension and the upcoming hoped-for approval of the finger-sized dry powder inhaler form of Tyvaso for patients with ILD, I'm very hopeful that tens of thousands of patients may benefit from access to the first targeted therapy for PH-ILD. A moment ago, I revamped the old saying about the road to hell being paved with good intentions, And I said, actually, the road to heaven was paved with good intentions. Perhaps an even better quotation, especially with regard to our development of Tyvaso for PH-ILD, comes from Bob Marley. Many of you recall his lines that if you know what life is worth, you will look for yours on earth. It is what all of us have done here on earth, starting with Sir John Vane in his laboratory, and then the ball being picked up by Professor Werner Seeger with his inhalation devices, and then, most recently, as shown by the authors of recent NEJM article on efficacy of Tyvaso for patients with ILD and PH, as shown in our INCREASE clinical trial, that we have shown great respect for what life is worth. The gap in these two Kaplan-Meier curves represents a horrible loss of life. I am very excited that on this personal journey through PAH, initiated by my daughter with Group 1 PAH, that we have been able to create more of a heaven on earth for the tens of thousands of patients with a different disease, Group 3 PH, that is ILD with PH. I look forward to closing the gap between those two curves and then moving both of them further upwards. To solve the problems of lack of systemic delivery with inhaled treprostinil and painful or problematic risks of parenteral systemic delivery, we next developed Orenitram, an oral form of treprostinil. This product was also our first opportunity to show that treprostinil dosed orally and on top of background therapy actually reduced morbidity and mortality from PAH. While I thought we had finally achieved the holy grail with this product, it had one annoying problem. It is hard to tolerate an adequately, aggressively up titration of these pills in a prostacyclin-naive body. The importance of this I will show heuristically in the following potential energy curve. We can think of pulmonary artery pressures, or equivalently, pulmonary vascular resistance, as an energy system in which the cardiopulmonary hemodynamics are at equilibrium in a stable energy well, or at disequilibrium in an unstable runaway state. PAH patients generally die from right heart failure, so it is really all about the right heart afterload in the final analysis. Right heart pressures, measured indirectly via mean pulmonary artery pressure, or PVR, are clear and cogent ways to assess disease progression. There is plenty of data from over 20 years that longevity, that is, survival, varies inversely with mean pulmonary artery pressures, especially above the mid-range of 35 to 45 millimeters of mercury. Arrow number 1 indicates factors that have pushed a patient out of a stable equilibria in which no treatment was required. We are ignorant as to the molecular biological details of what many of those factors are, but they could be any number of idiopathic or secondary factors increasing inflammation in the precapillary pulmonary vascular bed. This new dynamic catapults the patient over the energy well into the mid-range of PAP pressures. Human physiology has a second cardiopulmonary energy well in the range of 20 to 40 millimeters of mercury, which is useful for brief excursions of high altitude or other kinds of stress. However, if the forces indicated in arrow number 1 are chronic and not relieved, they will push the patient out of this second energy well into the unstable zone. However, if the forces of arrow number 1 are chronic, the patient may be maintained indefinitely in the middle energy well so long as they are managed appropriately with PAH-approved therapies as indicated by the triple-headed arrow number 2. The actions of these drugs countermand the actions of arrow number 1, but if these therapies are not managed or utilized properly, or if there is a lapse in compliance, then the patient may catapult over to the unstable region at the far right of the chart, as indicated in the right-facing arrow number 2A. Once the patient is in the unstable region, they may not have long to live. However, very aggressive treatment with parenteral prostacyclin indicated by the fat arrow number 3 may be able to push them back over the wall of the energy well and into the stable middle energy well of 20 to 40 millimeters of mercury. Once they are there, they may be able to transition off of the parenteral prostacyclins and transition to oral prostacyclins at an equivalent dose. Unfortunately, the tolerability of PK profile of oral prostacyclins do not currently permit starting patients at this dose if and when they are caught in the unstable region. We are now in phase 1 trials of a once daily oral prostacyclin we call Orenapro, with a more favorable PK profile, so that might be an option in lieu of parenterals in the next few years. I want to reemphasize that the chart is heuristic and exemplary, that the boundaries between energy states are fuzzy, that, of course, every patient presents with unique physiology and biochemistry, and thus every case is different. But, in broad brush, my personal journey with PAH and its treatments has led me to believe that the chart accurately depicts the basic reality. This video demonstrates the heuristic cardiac afterload model I just explained.
Afterload reduction to improve right ventricular structure and function in pulmonary arterial hypertension. In pulmonary arterial hypertension, PAH, distal pulmonary arteries narrow and stiffen, leading to increased right ventricular afterload. RV afterload is measured by pulmonary arterial elastance, estimated by mean pulmonary artery pressure, MPAP, over stroke volume. The thin-walled right ventricle, RV, is unable to cope with high pulmonary artery pressure for an extended period. In response to elevated MPAP and pulmonary vascular resistance, PVR, over time, the right ventricle thickens, becoming large and dysfunctional. The RV volume increases and ejection fraction declines, eventually progressing to right heart failure and death. Lowering MPAP to near normal levels may reduce hemodynamic stress, restore RV function, and shift RV-PA coupling towards normal, which has been demonstrated clinically after calcium channel blockers therapy for PAH and balloon pulmonary angioplasty therapy for inoperable patients with chronic thromboembolic pulmonary hypertension. Similarly, almost any right ventricle will recover within a few weeks after lung transplantation. Clinical study data from several retrospective single and multi-center studies suggest it is possible to near-normalize MPAP and PVR with early and aggressive prostanoid therapy in patients with PAH to achieve right heart reverse remodeling, PAH risk reduction, and long-term survival beyond 10 years. Afterload reduction to improve right ventricular structure and function in pulmonary arterial hypertension.
This data from Dr. Tokunaga illustrates well the point just made in the heuristic chart. The magenta line shows a slow increase in epoprostenol parenteral delivery, and the green line shows a rapid increase. Clearly, the rapid increase makes much more of a difference, and that kind of aggressiveness in parenteral treatment may be helpful to get patients out of the unstable region to the far right of the PAH potential energy curve. Because treatments for multiple pathways may be necessary for patients presenting outside of a stable energy well, that is, above 40 millimeters of mercury, and may require parenteral therapy, we have developed a protocol. We call the protocol the Artisan Protocol, which stands for Afterload Reduction to Improve right ventricular structure and function. The goal will be for this protocol to start with a simple, easy to use version of a parenteral prostacyclin pump, what the FDA approved last year under the name Remunity pump. The reason for this is that shockingly, over half of PAH patients who die without parenteral prostacyclin. No doubt they are likely in the unstable portion of the PAH energy curve on the far right, and nothing other than parenteral prostacyclin can be dose titrated enough to try to push them back over the energy wall into a stable equilibrium. Yet, when confronted with the complexities and dangers of current parenteral prostacyclin pumps, a large proportion of the patients demur. Unfortunately, this will all too often result in their deaths. Equally problematic is that when they are put on the pumps, they are often not adequately dosed. I believe if they were given a small, simple, safe, and easy to use pump, which is what the Remunity pump is, and if they were told that they have the potential of only being on the device for 6 to 12 months before being transitioned to a pill, then that artisan protocol would sound attractive and many more lives could be saved and extended. The Remunity pump looks like a smartphone type of device and is controlled wirelessly. The patient does not have to fill it with drug other than snapping in a pre-filled cartridge. Remarkably, the Remunity pump is the first drug pump in history that has no moving parts, none at all. A nitinol wire expands and contracts in response to electronic signals based on continuous subacoustic sensing of the volume of drug in the cartridge and the programmed flow rate. One of the most important parts of my current journey in PAH is to fill the cartridge with a form of treprostinil that will not cause pain upon subcutaneous delivery. This new product, which we call RemoPro, is due to go into human testing in the near term. While there is no correlation between site pain and delivered dose for the current Remodulin subcutaneous delivery system, unfortunately, many prescribers and patients believe that such a correlation exists, and this results in an underdosing of Remodulin. Hopefully, the Remunity pump filled with the RemoPro painless drug we are working on will enable more rapid acceptance of the artisan protocol. Let me share some data from other scientists that support my enthusiasm behind the artisan protocol. First, here are a couple of charts from Dr. Ishiguro noting the stark difference in survival between patients in the stable and unstable portions of the PAH curve I showed heuristically. Of course, there is nothing magical about 40 milligrams of mercury versus 42 milligrams of mercury, and similar data would result if PVR or biochemical markers of heart failure, such as BNP, were plotted in lieu of mean pulmonary artery pressure. The point is that while patients can live for a long time with right heart afterload pressures that are about 2 or 2.5 times normal, provided they are managed appropriately, but once the right heart afterload gets to 3 times or greater than baseline healthy normal, the prognosis is exceedingly bleak. This chart from the Vienna database shows a similar picture. The more successful we are at reducing mean pulmonary artery pressures, the longer patients may live. The top quartile of survivors are separated from the bottom quartile by a whopping 25 millimeters of mercury mean pulmonary artery pressure. These charts from Dr. Matsubara present a similar picture. His patients live indefinitely so long as mean pulmonary artery pressure is controlled below about 40 millimeters of mercury. One of the earliest steps in my personal journey through PAH was when I funded Drs. Norbert Vokel and Ruben Tudor at UCHCS to create a glassed-in ex vivo model of a tiny section of the precapillary vascular bed. There was unmistakable evidence of changes in cell morphology as a result of increased pressures in this closed system. Of course, This same change in cell phenotype and remodeling has been seen many times with explanted lungs from PAH patients who receive lung transplants. As I noted earlier, it is one thing to believe in the artisan protocol's hypothesis that substantially greater longevity will result from aggressive dosing of parenteral prostacyclin for patients presenting with a mean pulmonary artery pressure above 40 millimeters of mercury until their pressures drop below that threshold, and then transitioning them to oral prostacyclins for long-term maintenance. But it is another thing to prove it. This chart of the effect of PAH therapies on RV afterload reduction over the past 30 years shows very rarely have PAH agents achieved a reduction in RV afterload to the desired level, and almost always parenteral therapy was required. Hence, we have decided to conduct a study to test the artisan protocol, which we call the Artisan Study. It will explicitly test whether aggressive dosing of Remodulin to achieve a targeted reduction in mean pulmonary artery pressure below 40 milligrams of mercury, followed by a transition to Orenitram and other oral drugs for long-term maintenance, is associated with improved RV function. The new Remunity pump will be used in the Artisan Study. While we can all take a lot of satisfaction from improvement in long-term survival for patients with Group 1 PAH, it is obvious that the situation is still far from satisfactory. The French cohort data, shown above, is impressive with its nearly 25% improvement in 3-year survival, from 58% 2 decades ago to 73% more recently. But this is not good enough. This is no, in Bob Marley's words, heaven on earth. And we all know that we can do better. I am hopeful that the Artisan Study will show that the Artisan Protocol can make a positive difference in this regard. However, as you'll recall from the start of my journey, it had 2 main prongs. The first was a better pharmaceutical treatment for my daughter and others with PAH. The second was to cure the problem with an unlimited supply of lung transplants, not only for PAH patients, but for all patients with untreatable end-stage pulmonary disease. As with our drugs, the road has been long and windy with unexpected twists, turns, and triumphs. That journey has gone from pigs modified with 10 genes to knock out or knock in biochemistry relevant to immunology and hematology, including induced immunochimerism via concomitant donor thymus transplant, to manufacturing rows of lungs from billions of endothelial, epithelial, mesenchymal, and stromal cells, both separated by HLA class and also dedifferentiated from iPSC cells derived from a single patient, both streamed onto decellularized GMP porcine lung scaffolds and onto 3D bioprinted scaffolds from inert collagen-like peptides. In fact, for the past few years, we have produced over 500 lung scaffolds a year and countless lung cell types. Last year, I took out a full-page ad in the Journal of Heart and Lung Transplantation offering these scaffolds at no cost to any academic scientist with an approved research plan. Here you see me inspecting our lung manufacturing facility in Research Triangle Park, North Carolina. Essential to our efforts to manufacture hearts and lungs has been the guidance on our scientific advisory board for over 2 decades of Sir Magdi Yacoub, shown here with our partners Marianne and Bina. When others discouraged me from taking on such a moonshot, Sir Magdi encouraged me, describing the many moonshots in cardiopulmonary surgery that he has accomplished throughout his career. When others questioned what type of organ failure indications would be susceptible to clinical trials with a manufactured organ, Sir Magdi's immense knowledge of organ transplantation always provided us with logical clinical trial protocols, which we are now preparing to undertake over the next few years. For example, for our manufactured hearts, we are looking at patients with biventricular heart failure and ventricular access issues inconsistent with VADs. For our manufactured lungs, we are looking at patients who would have insufficient gas exchange capability after necessary removal of one or more lung lobes, which we would replace with our manufactured lung lobes. This chart provides an overview of the full array of our organ manufacturing activities. If you ride the ski lift up from the lower left-hand corner up Regen Mountain, then you can first stop at our EVLP or ex vivo lung perfusion slope. There, we have already prolonged dozens of patients' lives, many with PAH, by providing them with restored donor lungs that were initially discarded for transplant due to their poor condition upon donor death. We now operate EVLP facilities in Maryland and at the Mayo Clinic, as well as being in close partnership with Toronto General Hospital, where the procedure was invented by Dr. Shaf Kashafji. If you continue up Regen Mountain, you next get to our double blue square GMP scaffold and the recellularization facility in RTP, North Carolina. This is the place I showed you the picture of on the previous slide. We are hoping to commence clinical trials with these lung lobes by 2025, match 2 patients by HLA type after demonstrating enduring safety and efficacy in a human lung to pig transplant model. Continuing up Regen Mountain, you finally get to the Black Diamond Slope. This one involves autologously cellularized 3D bioprinted scaffolds, a double challenge for technology, but one that builds upon our allogenically cellularized D-cell scaffolds and our day-in, day-out work with EVLP lung restoration. Clinical trials of these lungs will begin sometime in the mid-2020s. Finally, on the right side of the slide, we also have some fun skiing on Zeno Mountain. Here, as the name implies, we use genetically modified pigs as organ donors not only for lungs, but also for hearts and kidneys. Each of these technologies are bridges to better outcomes for patients. Just like our pharmaceutical strategies are bridges to better outcomes for patients, and ultimately for a tolerable organ transplant as a definitive cure. The lesson of my personal journey is that many times the answer you are looking for is right in front of your face. Remodulin was the first medicine we made. I mistook it for a destination when instead it was meant to be just a bridge. It is meant to be a bridge to a more convenient form of therapy, such as Orenitram, which can also be a destination for some patients, but at worst, I hope it is also a bridge to a tolerable lung transplant as a definitive cure for PAH. Along the way, we learned that Tyvaso was an amazing treatment for PAH, even though it was naysayed by everyone from the Nobel laureate and my good friend Sir John Vane to all the pharmaceutical executives from Burroughs Wellcome and Glaxo who joined me to develop Remodulin. And not only has Tyvaso been a godsend for many PAH patients, it has also opened up an opportunity for treatment for tens of thousands of people with PAH-ILD and will be further studied for its potential application in patients studying from idiopathic pulmonary fibrosis. You will of course notice that we have always followed multiple pathways to unlocking our goals. Biochemistry is at least as complex as anything in astrophysics, and that is saying a lot. So, disappointment is inevitable, and the only way to increase one's odds of success are to always place multiple sound, well-reasoned, well-executed shots on goal. As my hero Robert Heinlein says, of course the game is rigged against you, but if you don't play, you can't win. Us mere mortals trying to outwit millennia of natural evolution is a damn tall order. But if we don't try, and try smart, then we can't win. So we've got to try with all the intelligence and initiative that we've got. In conclusion, I want to thank the fonts on my own initiative and intelligence in this PAH game. my daughter Jenesis, and my partner Bina. Jenesis, now in her 30s, works at United Therapeutics as our digital media manager and head of telepresence activities. I would also like to thank all of my teammates at United Therapeutics for this presentation and for the development of the Artisan Protocol and the Artisan Study, and for so very much more. I want to especially acknowledge the crucial roles of Dr. Yan Liu and Dr. Gil Golden. My journey was to help our daughter Jenesis enjoy a happy life. She has and is, but now that I've come back around to where I started, I realize that when you make one person happier, you can make a world of people happier. Saving one life is, in a very real way, saving the world. Thank you.